Hello students, today we will see haemopoiesis. So hem means blood, poiesis means production. So haemopoiesis, it is development and maturation of the cells, that is RBC, WBC and platelets in marrow, bone marrow. It is called as haemopoiesis. There are three types. Erythropoiesis, it is development and maturation of RBC. Leukopoiesis, it is development and maturation of WBC. And thrombopoiesis, it is development and maturation of platelets. So in erythropoiesis, it takes place usually in bone marrow. So first cell is hemocytoblast. So this is most precursor cell. It is most larger in size, round cell with nucleoli, the bluish purple color cytoplasm and nucleoli present in the nucleus. Then it develops into the pronormoblast, then early normoblast. It is basophilic, that is purple in color. Then intermediate normoblast, it is polychromatic, that is cytoplasm is purple and pink in color. Then late normoblast, it is around 7 to 10 micron in size. Nucleus is eccentric, nucleoli absent and cytoplasm pink in color. Then the nucleus breaks down and then it is formed, that is reticulocyte. This is immature stage of RBC. Nucleus absent but nucleus present in the cytoplasm in network of the basophilic ribonucleoprotein. It is called reticulocyte. And then finally the mature RBC develops, that is 7.2 micron in size, nucleus absent and cytoplasm is uniformly red with the central pallor. So in this diagram you can see all the stages of the erythropoiesis. Thrombopoiesis, it is maturation and development of the platelets from the precursor cell, that is hemocytoblast. Then it develops into the megakaryoblast, which is 20 to 25 micron in size, then promegakaryocyte and then megakaryocyte. It is 10 to 14 micron in size. It is irregular in shape, nucleus in form of many lobes, nucleoli absent, cytoplasm pale blue and granules purple blue in color. And then from megakaryocyte platelets develop. It is also called as thrombocyte and the size of platelet is 2 to 3 micron, non-nucleated, round to oval bodies. Cytoplasm is blue or purple and platelets are formed by nipping of the pseudopodia of the megakaryocyte. And the significance of megakaryocyte, if it is increase in bone marrow cells, then it may be the condition like thrombocytopenia or idiopathic thrombocytopenic purpura. Leukopoiesis, it is maturation and development of the leukocytes, that is all the cells like neutrophil, eosinophil, basophil, then lymphocyte and monocyte. So it is usually seen in bone marrow, lymphoid series seen in lymph nodes and monocyte in spleen and bone marrow. The stages are, the first stage is hemocytoblast, that is common stem cell. It is larger in size. Then it develops into the, matures into the myeloblast. It is 10 to 18 micron oval, big nucleus with nucleoli, and cytoplasm is usually light blue, no granules. Then promyelocyte, it is differentiated into neutrophilic, eosinophilic or basophilic according to the granules present, either pink, orange or bluish black in color respectively. Then it matures into the myelocyte, which is smaller in size. Then eccentric oval nucleus is present and then no nucleoli, granules in cytoplasm are present. Then it develops into the metamyelocyte. The nucleus is kidney shaped, cytoplasm granules present, and then it develops into the band form, in which nucleus is in band shape, and then finally segmented nucleus of 2 to 3 lobes nucleus is developed. So this is all about the neutrophil or eosinophil or basophilic leukopoiesis. Then lymphoid series, it is hemocytoblast, develops into the lymphoblast and then finally mature lymphocyte. So lymphoblast is round with the bluish cytoplasm and nucleus which contains nucleoli. Then monocyte series, hemocytoblast, monoblast, it is size of 14 to 20 micron, nucleus is oval or kidney shape, nucleoli present in the nucleus, and cytoplasm is bluish gray. And then finally monocyte, that is nucleus kidney shape, nucleoli absent in monocyte, is produced. In this diagram you can see all the stages of the developments, like in neutrophil it is neutrophilic myeloblast, then promyelocyte, myelocyte, metamyelocyte, band form, and finally segmented neutrophil. And according to the color differentiation in the cytoplasm, you can differentiate pink color neutrophilic, orange color eosinophilic, and bluish black color that is basophilic. So hemopoiesis, if it is seen normally in bone marrow, or lymphoid series in lymph nodes, and monocyte series in bone marrow or spleen, then it is called as medullary hemopoiesis. And in pathological condition, if the hemopoiesis seen in other organs like spleen, lymph node, then it is called as extramedullary hemopoiesis.
Hello students, welcome to this session. So today we are going to discuss about bone marrow examination. Structure of bone marrow. Red marrow present between bony trabecula, which contains cells of myeloid, lymphoid, erythroid and thrombopoietic series, and reticulum fibers, blood vessels and fat cells. The examination of cellular composition of bone marrow is essential for diagnostic purpose in wide variety of disorders of the hemopoietic system. The types of bone marrow are bone marrow aspiration and Triffin biopsy. Bone marrow aspiration, it is aspiration of particles of bone marrow with help of aspiration needles. The needles should be strong, wide bore, and it should be with the guard to prevent over penetration. The examples of bone marrow aspiration needles are Klima, Salah, Jamshidi. The sites of bone aspiration collection is in adults, body of the sternum or iliac crest. In children, if it is less than 1 year, then it is medial aspect of the tibia, and for more than 1 year child, it is iliac crest. Now we will see procedure of bone marrow aspiration. So in that, first local anesthesia is given into the periosteum, or sedation or general anesthesia is given in children. Then clean the parts, like if you are taking for adults the body of the sternum, or for the child medial aspect of the tibia or iliac crest, then clean the parts with aseptic precaution with spirit and betadine. Then bone marrow aspiration needle is introduced into the bony cavity by to and fro movement. After cavity is penetrated, stylet is withdrawn and tightly fitting syringe is attached. Then brief and strong suction, you can take 2 ml of bone marrow tissue. Then films are prepared immediately by placing the aspirated material on glass side and sucking of the blood which is contaminated. Then air dry, and then films are fixed into the spirit or methyl alcohol, and then you can do staining of the smears by Leishman or Wright stain. So by Leishman or Wright stain, you have to add the stain for 5 minutes, then add buffer for 5 minutes, and then wash with water and dry it and see under high power and then oil immersion lens. Now after the smears are stained, then we can do examination of the bone marrow. So in that we have to see certain points like cellularity, so either normal or hypercellular or hypocellular. So that we can make out by the cavities which are present because of the fat cells. So if the normal cavities are seen, then it is normal. If the cells are more and cavities are less, then it is hypercellular. If the cells are low and cavities are more, then it is hypocellular. Then we can study erythropoietic series, leukopoietic series, then plasma cells which are seen in increased quantity in multiple myeloma, megakaryocytes, if they are increased, then they are seen in thrombocytopenia. Parasites like blood parasites like Plasmodium species or the Leishmania species we can see, and then we can do special staining of the bone marrow examination for identification of iron. We can do it by Prussian blue, or for the acute leukemia we can do special staining like periodic acid shift, peroxidase or Sudan black stain. In this picture we can see all the myelopoietic series like myeloblast, myelocyte, metamyelocytes, band forms, or erythropoietic series like erythroblast, normoblast, reticulocytes, and thrombopoietic series like megakaryoblast, megakaryocyte, and the platelets. So in this picture you can see examination of bone marrow. So all this series of cells of the myelopiesis, erythropoiesis, thrombopoiesis and the blood parasites like microfilaria, plasmodium species or leishmania.
Hope you find this session useful for your understanding and for your exams. Thank you.